Okay, how many of you have flights uh, in a few minutes, hours? Okay, feel free to walk out whenever you need to. Um, Thank you for having me and my group. It's just been absolutely incredible. Um, I think there are still some, oh, I'm hearing myself here. Still some things, even after 4 or 5 days of discussions that haven't been touched. So I'm going to try to briefly jump through some of them, and it might feel like a soup of ideas, but I'm around for a little while, and we can talk a little bit later afterwards too. Um, so first of all, I think one of the most important things when I'm thinking about in science is the historic context. Uh, Although I was not present at this very famous meeting in 1981, I was a 1-year-old child at that time. But I am sure many of you in this field recognize this photograph. Uh, the lots of big ideas around, the energy usage in computing, the idea of reversible computing, all discussed at this meeting. I think in some sense, these types of places do remind us that power of a community itself when we come together from many different fields. Uh, I started my journey in grad school thinking about a strange idea, which is I wanted information processing to not just compute with information, but to build. I was excited about materials. I wanted, inspired by some of Eric's work and others, that led to this funny little place in a kind of a historical context of making computers out of bubbles. Show that low Reynolds number hydrodynamics can actually be Turing complete, and you can actually exploit nonlinearities. And since a lot of you talked about resistor networks, I want to give you a little quiz. This is at the end. So I have flow. You can see two little channels going by. You can see the funny adder-like structure. I'm going to pump these tiny little bubbles through them. And one option is nothing would happen. As they arrive, they leave it the same way. The other option is they talk to each other. I'm just curious how many people think nothing is going to happen as I play the movie? So I'm talking about timing of these. They are traveling. And difference here is that we talked about nonlinear resistor networks. Here, the nonlinearity actually travels through the channel. So how many people think nothing is going to happen and the delay remains the way it is? Okay. And how many people think that these bits will talk to each other and synchronize? Many of you have read the paper. They actually do talk to each other and they synchronize. But here's the puzzle. This is Stokes' flow. If I played the movie backwards, what should have happened then? They should unsynchronize, but then who should go forward? That doesn't make sense. That's paradoxical. I've actually shown you something quite funny here. It requires a lot of thinking in what is going on in this system to be able to say what actually happened. And both camps were right because I didn't actually tell you the boundary conditions. When the boundary conditions are not symmetric, they will synchronize. But when the boundary conditions are actually symmetric, nothing will happen. So anyway, just history of nonlinear networks. We're actually thinking quite a lot about to now implement funny things in these. Let me skip this. Okay, I went to the dark side after finishing my PhD, and I just got excited about our natural world. So much of what I'll talk about today is not too much from the perspective of what all of this can do to computer science or to our technology. Can we think computing as a framework of thinking about biological problems? We have collaborators in plain sight. What I mean by that is an incredible diversity of organisms that are out there that we could learn from deeply in trying to understand the class of technologies that we're possibly excited about. Okay, so another historic fact, Jennings in 1906 published a very important book. It was almost, he was put out of the community right away because of this book. He said, behavior of the lower organisms. What he was trying to say that individual cells can think and they can learn. And he did remarkable sets of experiments to actually show, for example, stentors could learn and single cells could learn. But, this was the era of people were starting to think about neuroscience And he was actually put aside. People said, oh, his experiments are not repeatable. Really, it was not a good ending from a perspective of championing the lower organisms. I am going to champion the lower organisms today. Ironically, some of the students that followed took this notion that, since Cajal had drawn these nervous system drawings, and that led in single cells this idea of neuromotorium. People started seeing cytoskeleton and thought that that's the equivalent of the nervous system. And inside the cell, people started to look for the brain and started to label these sets of things. And it's so ironic that almost 100 years later, we have understood that cytoskeleton can learn and cytoskeleton do really remarkable things. But this time, once, after 1950, some of this was published, a couple of experiments came along, and all of this was thrown away. Frankly, that had the dark history to a point that nobody wanted to touch the idea of learning and adaptation in single cells and non-neuronal systems for a very long while. Anyway, I think we're going to revive a little bit of that, but this history is both dark and fun. The other thing that hasn't been brought about, although embodiment has been talked about, is just this beautiful, it's called synthetic psychology. We can do thought experiments in the space itself. If you haven't read this book, just a remarkable book to read. I will talk about this in the context of what I care about, which is how do we even classify some of these ideas? So, for me, I already highlighted, for me, computation might be a good language to be thinking about dynamics of living systems. And then naturally, in these sets of systems, sensation, memory, computation, learning, actuation, all combined together in this framework of what I'll call perception action machines. This is an old idea. It's been talked about by many others. We've been thinking about this, and all the work that you see, you should be thinking about the world layer, the sensory inputs coming into the internal states. The internal states can essentially change themselves. You act upon the world, you can actually change the world, and this cycle continues in some way. And actually, we've been thinking about philosophically extending these notions of perceptive machines. Why should we stop here? Been thinking about reflexive machines, perceptive, attentive, cognitive, learning machines, which a lot of people talked about, exploitative, and also curious machines. And these sets of classifications are just for fun, but it allows us to be thinking about the natural biological dynamics that we see. We have some rigorous definitions for each sets of these machines. But again, this is just our own internal classification because it helps me when I see dynamics of an organism, how I might be thinking about it in terms of classifying them. So, okay, now with this insight, let's talk about a few sets of physical systems. I'm going to spend only 5 minutes on each one of them, but I just want to give you a flavor of how to think about these ideas in real physical experiments. Let's begin with the dream that I had. Computers should make stuff. And I'll talk a little bit about this framework of an organism that builds things, I believe, using algorithms that I don't yet understand. This photograph that you're looking at is something Santa Barbara on a beach. I picked up something that was crawling around, and I saw something like this. It looks like medieval weapons. And what this is a biological organism programming this shape over and over again on its skin. What you're looking at is a single crystal. So it's an ossicle mineralized in this structure by some process. And again, this is calcite. In its crystalline form, it looks to like if I do this in a dish, this is what it'll make. But the organisms, and many organisms, including many echinoderms, actually make structures that are these beautiful organic structures. The question is, how do you shape crystals? This is a long-standing question in biomineralization, but we've learned something new about this idea, that partly it's not just about the molecules at the lattice structure that actually shapes this. So this is the puzzle. How would you, by looking at these things, derive algorithms, what this might actually have to do with computing to begin with? There's a lot of work in mineralization. I'm only going to talk about at this scale. There are a few cells collaborate with each other to build something new. So here is that little organism I talked about. If you love tardigrades, this is the new tardigrade. It's very cute. But you notice that it is armored. It has all these little sets of structures. And the reason we love this, the fact that this production is happening in different parts of the body over and over again. I can do perturbation experiments, I can do control experiments. And this work is led by two graduate students that are here, Pranav and Charlotte, with a couple of collaborations. So let's just look at the system. There's a huge amount of diversity of these sets of shapes. Started wondering what could be programming these, and we decided to essentially map all of them. So this is the mandala of all these crystalline structures that the single organism built. And, of course, for some of you who are excited about topology, this is a fairly complex topological structure, but it can actually be described well. We love this idea of a lab on an animal because what is it that's so different about neighboring structures that are growing? What is similar? We can do certain sets of statistics. For just today, I want to show you one little data point on this teasing of an algorithm on this number 5, which we call a table ossicle, which is the most abundant ossicle in this. So first of all, you look at this, you start seeing symmetries. There are different sets of symmetries, and those sets of symmetries might imply that there was a seed. Just looked at these sets of shapes, and we started searching for seeds, and it turned out we found seeds. The structure is tunable, which is quite exciting because it is a crystalline lattice. It's been built, but it actually does take some local information. Most of this is internal. And again, we can analyze these sets of structures for all kinds of topological properties that I don't have time to talk about. There is a certain class of lattice that's being programmed. And the question is, what sets of rules can program these lattices? And then just to prove to you that nature sometimes is digital, it truly is a single crystal. Taken that ossicle, when I grow something on it, you can see the crystalline axis is actually pretty visible. So here was the magic moment. We figured out, let's just watch this thing. And we were able to take this poor animal, hold it down for many days to watch the ossicle form in real time. And you're going to see the seed of the algorithm there. You can see tip bifurcation. Seeds that grow bifurcate at tips. Actually, in the next movie, you're going to see this fusion event. Somehow these tips are talking to each other, and then they fuse together. And somehow that process continues over in some sense to be able to build this structure. Now, I think I'm going to skip much of this. There is a lot of work that we have done now to really understand the scaling laws, but I'm just going to show you the key actor, the key puzzle in this. What's really happening is how can cells know what they are building? And the key answer lies to the fact that when we label the sets of cells, the cells that form, this is 4 or 5 cells that build this structure. We don't believe that there can be some amount of information, but much of the construction is coming from these 4 or 5 cells. They wrap around this rock like a saran wrap. The feedback loop here is that the object that you are building, you become that object. The shape of the cell and the shape of the ossicle is actually one and the same thing. And then there is a 40 nanometer thin little wrapping that you can actually see in that other picture. This is a TM image of this, where these cells wrap this around and secrete in this in situ calcium and many sets of proteins in that allows the structure to grow. Now that structure grows and actually gives feedback to the cell for what it has built, and then this process continues. Now there is a lot more to this in terms of transports and networks, so I just want to show you one network. First of all, much of the growth actually happens on tips. How do they know where the tips are? There's lots of protein components. This is the notion that I told you that tips actually talk. You can see this transport on the surface, and then the best picture to show here is right here. This is the internal transport network. Very much like Karen had talked about transport across sets of networks, now these are microtubule networks that act as highways for these sets of organisms, the sets of cells, to put materials in certain locations. And for those sets of rules, you can essentially derive the kinds of structures that this might form. And to give this context of as we play with these sets of transport rules, we find that there is a certain class of programmability associated with this. Of course, I have not touched at all about what's happening at a molecular scale, which lots of people in biomineralization work. But doing this work, what we have realized is the fact that the cellular and the physiological context controls a significant part of the structures that many of these lattices will form. Rather than if I was to take all those sets of biomolecules and put them in a reactor, they might not form. This, we believe, is probably one of the reasons biomineralization as a field has been stuck, because we've been only thinking about bottom-up assembly, but it's both bottom-up and top-down assembly. That's all I want to say about the system. I'm going to jump to tell you about now something completely different, but it was, oh, I think here is a puzzle, just if anybody has ideas. Just like tree crowns, many of you know that trees that grow close to each other don't entangle. Specific species of trees do that. They have a sense of self. So ossicles that grow close to each other don't just entangle and wrap around each other, they stop. You have no idea, I mean, sets of cues possibly, but they do fuse with themselves because, as I said, the two tips fuse. Clearly, there must be that the clusters have identity. Otherwise, this should actually not be possible. We have no idea how this is done. Okay, so no, actually, let me tell you one more quick story on the cellular context, which has to do with dynamics quite a lot and behavior, and then I'll end with some of the elastic networks work that we've been doing. The other thing, bringing back this idea of cells and learning and behavior, I want to start with geometry. I think geometry is often thrown out of the box sometimes. This is just a classic picture of a few beautiful eukaryotic cells. What I love about this is just you see this immense diversity of geometry, but I have no idea what this geometry is doing. So how do I think about function and geometry and connect that to dynamical systems possibly? We pick one of these organisms. This is called lacquery. And I want to show you a new result that we have just found in the system. I go muck around in ponds with all my students. So we literally find these things on scum. Worth doing. But this is, when you get it right, this is what you see. What you're watching is a single cell. And it's a hunter. And it's going to, at some point of time, I want you to be thinking about where is the algorithm? What is this guy thinking? And wait for a second. It is a movie, so I know it's about to happen. By the way, this is not a worm. It is a single cell. And right there. That's a catch. So this is, I'm waving the thing around. It's going to engulf this thing and continue this process over and over and over again. Now, again, it's the same single cell, 40 micron in size, but now, like the movie Incredible, the arm is actually a millimeter long. It's the same cell that has now trapped that. First of all, some conservation laws kind of get into question. Where the hell did all this material come from? How do I control this? Where is the controller? So, and I think, we figured out how to grow this cell. It was a pain. But once you do that, they are not cannibalistic. They do not eat each other, which would have been interesting and fun by itself. They also don't form knots, which we had also thought quite a lot about. Like snakes don't knot themselves unless they're infected by a virus. So now the question is, what is really going on? How do I think about activity, dynamics, and algorithm in this context? Something very interesting. Many of you in mechanics notice this seems like a buckling pattern of some kind. There is surface activity, which is cilia, all this cilia on the tip, and you will see that same process play out again. This is all real time, by the way. This is the surface activity. Now, the surface has a tip activity and all throughout the neck, and we started thinking about this. The first question we asked ourselves is, what is the search algorithm? Given no information in the environment, we found that it has a very comprehensive search around it. It doesn't have blind spots. So the tip, if I map where the tip of the head is as a function of time, it covers its surrounding and it seems really random. That we got started thinking about chaos. Would it actually be using chaos? There are certain sets of scaling laws. So the neck, as it gets larger and larger in volume, your search space starts getting larger. There is a limit to this kind of an algorithm. But one of the key ideas was starting to use activity to program this. And I think much of this is really thinking about how do you forget? The mathematical framework for this is you have an elastic rod, activity at the tip that turns on and off. Now the activity is completely periodic. We can write down a non-dimensional number in the terms of elasticity and activity. And I'll just show you what it looks like. This is the full, both elasticity and hydrodynamics is solved because it's a coupled set of an equation. You noticed when we reverse the activity, there is a buckling. Now that buckling is really interesting because you could forget information in that process if that buckling could lead to certain class of a chaotic oscillator. And this is what Deepak was able to show. I'll show you this is one regime, elasticity and activity, and watch any of them and see if you can find a pattern. So say the one in the middle, dimensionless activity, do you see a pattern? Right there, it's a 6-period oscillator. Actually, it turns out it does form oscillators that are also not symmetric. Right there is a 10-period oscillator. It is going to every one of those 10 locations in a very particular sequence. But as we start scanning for parameter regime, we actually find majority of the parameter regime completely aperiodic activity. And now if you try to essentially predict any of this activity, I have to right there, you see now it's starting to comprehensively search space. And we have theoretically shown that this oscillator is chaotic. I don't have time for that. But I will show you that the experiment that you can do at home to prove it to yourself. Go water your lawn. You've been away for a few days. Use a flexible hose and turn the water pipe fully. You will notice that the hose actually starts oscillating. This was discovered for rockets because rockets were not stable. This is called follower forces because now this is a non-conservative force. Direction of the force in the system depends on the state of the system itself because the tip direction changes as your filament buckles. That's the key idea. But of course, nature is exploiting it. And one of the fun things I've been thinking a lot about is could we program? And we don't have a systematic way, but we've been searching using frequency modulation and local and global search to now start program this filament in ways and directions that I could tell some amount of it could be chaos, but some amount of it could be I really want to home the device, for example. Okay, and in the last 5 minutes, actually, no, I have to say something about conservation laws, because you can't come to a physics meeting and show something that seems to violate the conservation mass or material. It's the same cell that you are seeing that is forming. Sometimes I can't even measure. What is this extension? Where is this material actually coming from? This is all real time. I can't produce membranes. I can't make proteins. And we started actually after a lot of work, a fantastic graduate student, Ellie, we started thinking about origami. And it actually turns out now we've proven it's the first example of curved crease origami at a cellular scale. And the microtubules and the membranes are patterning in a manner. So this is sort of the cross-section of the membrane, and this is what you can think about what's happening at the membrane. The membrane is actually folded into a helical curved crease origami that now sequentially unfolds. So unlike a traditional spring, and I'm just going to give you, you're calling this lacrigami. The reason this is important is because the elasticity of this actually matters in the context of thinking about what will happen. So I'll just let that movie play. You're seeing essentially a cylindrical object that, as I point out, singularities, which are these D cones, folded to unfolded state, travel along the helix. So it's not like it's a non-affine spring in some sense, that when you stretch on it, it unfolds serially. Very much like when you throw out a fishing line, the spool remains the same and the line that goes out actually serially unfolds and folds. There's all kinds of really fun mathematical ideas hidden around here. Okay, in the last couple of minutes that I have, I want to tell you about, much of what happens is when we are thinking about biological sets of systems, we don't understand them, so we go back to the lab and build those sets of systems. Some of you have seen the set of a poster, which we believe we have stumbled upon a new mechanical object that I want to tell you about. This is work with Vishal, fantastic postdoc in Ian, and Vishal is in the job market. Pranav is also applying for postdoc positions right now. Okay, so I think I have to say, going back to applications and things, I want you to first think a little bit about your shoe. You walk around, imagine a structure that has the capacity to self-learn patterns that it finds around for certain purpose. And we started thinking about shoes for some reason. We all go hiking. Imagine your shoes adapted and upgraded to a given terrain if you walked in that terrain for a while. So input in this is essentially embodied environment. The system updates itself and changes to a certain function. And I think one of the key things that we care about in this is to do this all in an unsupervised, self-learning, completely embodied, nothing done outside, just a mechanical material itself. And this is what we want to make, quite literally. So 3 sets of things that we're excited about. System should be passive. I don't want batteries in my shoes. It should learn continuously. I go to a new place, it should learn something new. The way we started thinking about this, first of all, is actually this notion, what I'm calling a directed spring. It's a little bit of a strange idea that springs can have directions, and I'll explain what that means. So explore this. You've all heard a lot about adaptive springs. Springs, given a given type of an input, can change their stiffness. We're going to change it. The stiffness can go up, stiffness could go down. There is a threshold function, so they should hold on until the amount of activity around them increases, and only then change the stiffness. The key part is that there is a symmetry breaking in which the head and the tail, so only in a scenario where the amplitude at the tail is higher than the amplitude at the head, the stiffness should increase. But otherwise, the stiffness should remain the same. Now, this is very different from a diode. Forces are still being propagating just like a normal spring. There is a second time scale in the problem that learning or adaptation only happens in a directional manner. Fun hypothetical idea. The first thing we got inspired by was that, some neurons have directionality. Oja's rule, which I talked a little bit about, has directionality. Could we do that in a mechanical domain? Long story short, we were able to build something like this, and I'll just show you actually how the object works. This is the object. So first, just proof of principle. Forget about what's in there. Let's just watch it. I am shaking the same object on the tail end. There's a little loop, which is essentially an elastic ring. The shape of that is different, so hence its rotation by itself gives you different stiffness. But if I just take the exact same object, shake it on the other side, you see that the loop doesn't turn. So in this scenario, forces are propagating, but the update doesn't happen, while in the other one it does. First of all, how robust is something like this? And actually, I just want to show you that as this is updating, Ian can go in, mess around with the spring, it'll come back, and it locks back in again. So, of course, there is a dynamical system in there. It's not just a regular spring. But conceptually, I'm going to walk you through where does directionality actually come from. So you can think of it as 2 different oscillators. There is a pendulum, and then there is an oscillator, which is a 4-bar linkage, which is coupled to this rotation. And that rotation gives you the stiffness. I already mentioned that in a second. Of course, there is Coulomb friction that gives us the threshold. So the simplest way of thinking about directionality is if I have a pendulum hanging on another oscillator, which is a spring. If I pull this on the left, you'll see that that swings to the right. Here, by definition, tail and head is such that the positive axis pointing from tail to head. And if I do the same thing now for the object, same object, you'll actually see that the 2 sets of oscillators, which is the displacement oscillator for the pendulum and the stretch, are actually out of phase. So this is what we are truly exploiting, this in-phase, out-of-phase mode locking. We still have work to do because we have to take this asymmetry, use it to update. And this is where really the mechanical genius of Ian comes in. And I can tell you lots of rational stories of how we come up with these, but this is just staring at the sky sometimes. And one way of thinking about it is there are 3 sets of components. There is a pendulum, there is a ratchet, and there is a 4-bar linkage that's connected to this ring. And one way of asking this in the context of electronics is that the update only happens when the gate is open. The gate is the coupling between the 4-bar and the ring, and when the drive is on. When those two conditions are met, we can essentially do the update rule. What's very exciting about this is I'm not diving too much into this because this is only one tip of the iceberg of the kind of computations we can do in this set of a context. And we can rigorously prove that this is essentially directional. And I just want to show you just in the last few seconds, what could it actually do? What could we do with a system like this? So first of all, there is a physical realization of this system. We can show that computationally and experimentally the data match. I think of this much more in the realm of making circuits. These elastic circuits are topological. So a ring by itself, because these arrows matter, I often think of this as activation and inhibition. There's a massive class of circuits that you can make purely using a few sets of nodes. So if I was to change the arrow in that triangular circuit, behavior would be completely different. And again, we can not only just do these oscillatory, we also have a mechanism to actually do switching where the K just goes and latches to one max value or one min value. This is the circuit for that. I think it's too little time to actually explain this. Maybe I should conclude by showing that we can run just like the OJ's rule. We can essentially do PCA with this in an unsupervised kind of a manner. In this situation, our amplitudes are what our inputs are. This really does look like kind of the Oja's rule. And we can essentially do in this system an unsupervised approach where initially I'll get a certain sets of inputs. The K values would adapt to tell me what was happening with those sets of inputs. I give it a new input that changes. One complication is we can't do a multiplication term. So technically, we're not implementing Oja's rule. So there's still work to be done in terms of thinking about getting it actually there. Then I want to close with this. I had wonderful conversations with a few colleagues here who work in the electrical domain. We're still actually struggling whether there is an equivalence of this idea in the electrical domain. So I would actually be very curious. Carl thought that this has inklings of this QFP that they have been building. There are, of course, ideas around memristors in some sense, but it doesn't have the classic directionality that we've been thinking about. So now we are actually pursuing, trying to write down all the equations and find an electrical analog of this because it might be fun to build some shoes. Okay, I'll stop there.
Well, we've eaten into the question, but then there's a few questions. Does it have chemoreceptors at the edges of the?
Yeah, we did that experiment, and it's still work in progress. The data set that I showed you was trying to understand its behavior in a zero information environment. And then we still know it has a search algorithm. We do this kebab experiment, which is a skewered kebab. We take the organism that it eats, we put it in on a little pipette and feed it. And we see that its time to find it statistically on average does not change. So our field, we don't believe there are any chemoreceptors. It's a contact receptor. The moment it makes contact, we feed it beads now, so we can coat it with different types of cues to see what is it actually detecting. And there is some, I didn't talk about this, but there has to be information propagation from the head, the entire system. Because what if you just touch that favorite pizza, but then you moved again? If you don't shut down your dynamics immediately. You notice in those movies, the moment the contact happened, beam almost froze. And now we've been able to get some calcium signaling working in the system. So I do believe that there's probably a calcium signal that immediately propagates. Say, wait, you found something, pause. So that's all we know so far.
Yeah, so is there some insight from this previous organism you showed, which like wraps around and makes structures to be applied to this problem of a shoe?
Yeah, I think the insight for me personally in that space is I don't like this idea of this notion of, are we going to compete with the chat GPTs of the world? I want the computers to build our new world. I think there is so much in that space that I haven't seen. So I think one of the things that we study in this perspective is those are single crystal structures. They are using certain class of network and transport rules, but at that same time, their output is an object that I can hold in my hand. That's been a dream. And I didn't mention this, but we've been trying to do what Eric has been able to do in molecular systems, like build a Sierpinski triangle. They're culturing these cells, perturbing them to understand their rules. But I believe computers should, now it's time that they should make stuff, hopefully make themselves.
That the crowns don't want to make the monetization. What do you think is the evolutionary disadvantage for them to merge?
Very important. So actually, it's a beautiful, and again, there's a whole another talk that's hidden in this talk about evolution of the neuron that I didn't talk about, going back all the way to fossils. But one important aspect of sea, how many of you have held a sea cucumber in your hand? Some of you have eaten it too? Yes. The echinoderms are known to make these incredible, remarkable ossicles in their skin. Sea stars and sea urchins fuse all their ossicles, and hence when you hold a sea star, it's stiff. Somewhat, sea cucumbers figured out to not fuse their ossicles, and hence they are flexible. That's his key difference between this entire class itself, where in this case, what you're looking at, this is essentially sea urchin. The ossicles first formed independently. They didn't care about who's making what. You just made a jammed structure. What we don't know so far, and actually this is another work that we're currently doing, is I didn't say, what is the purpose of all this? What does it imply? I mean, for sea star, it implies defense in many ways. For sea cucumber, these things are fairly far away, as far as we know that they do not purely mechanically interact. This idea that I was talking about, the armor, may or may not actually be true. So in certain species, in echinoderms, there is fusion, and in this system, there's no, but it's a very puzzling question, because for those tips to fuse, for the structure to form, the cells are fusing. It's a synctium of cells. The cells form these actin filopodia that fuse together. That's what guides the branch. What's the difference between the two of these neighboring sets of cells that are growing, that are poking their branches into each? So how do you define self in a system like this at a scale that's smaller? And again, I think these types of ideas also occur in neuronal circuits. It's actually molecularly, there are similarities between patterning rules that are used here, the patterning rules that are used to make neuronal circuits. For example, the neurons in the skin of a Drosophila also show this crowning effect. They want to cover the area as much as possible, but not contact.
Thank Manu one more time.
One small practical announcement. I think